Hi students, today we will discuss genetic engineering. We know well this is a very new technology nowadays which we are implementing for crop improvement or to get maximum agricultural produce. Started from the work of Stanley, afterwards the work done by various scientists. Till today we are adding so many things. So in this plant genetic engineering, what we are doing exactly, this we will discuss in detail. So for this, we are taking DNA from the host plant. We are multiplying that DNA by considering various vectors. With the help of vectors, we are succeeded to multiply the gene which are going to be transferred from one organism to another organism. So in this technique, manipulation of genes, we are playing with the genes to achieve our success, to achieve our target. So in genetic engineering, we are getting the DNA from the desired characters, and that desired character, that particular gene, we are isolating from the host plant and we are incorporating into the plant which we want to have that particular desired trait or characters. So as far as applications of this genetic engineering is concerned, we can use this technology for the betterment of agriculture, to get various medicines, to increase the production of secondary metabolites or various characters. So with the help of certain animations, we will discuss in detail about the plant genetic engineering technology. So genetic engineering, also known as genetic modification, this is the direct manipulation of an organism's gene using biotechnology. As we know well, it involves the use of various techniques to alter the DNA sequence of an organism, allowing for introduction of new traits or characters. When we see the overview of genetic engineering, there are 5 main steps in the process of crop genetic engineering. Here you can see on the screen all these 5 steps I have shown here. As far as key steps of genetic engineering is concerned, so these steps are basically the same for all genetically modified crops, out of which the first step is isolation of DNA. The DNA molecule is isolated from an organism using various techniques such as PCR, which is also called as polymerase chain reaction, or DNA extraction. So the cutting the DNA, the isolated DNA is cut using restriction enzymes which recognize specific DNA sequence and cut the DNA at those sites. So here we are having 5 steps, already I have mentioned. The first step, extraction of DNA. Second step, clone a single gene. Third step, design the gene. After designing the gene, the fourth step, that is nothing but the transformation. And the last step, that is backcross breeding. As I have mentioned that, after cutting the DNA, insertion of foreign DNA is very important. So a piece of foreign DNA known as a transgene is inserted into a cut DNA molecule using the enzyme called DNA ligase. So here, this is the cell. So every organism has a molecule called DNA. So this DNA is present in the cell. In every cell that holds the information for how to produce all the proteins that organism will need during its entire life. So all of the DNA is extracted out of an organism that has the desired trait. Afterwards, formation of recombinant DNA is very important. The resulting DNA molecule which contains the foreign DNA, which is known as recombinant DNA. So here a single gene That codes for the desired protein must then be located and copied out of all the DNA extracted from the organism's cell. Once the gene of interest has been cloned, the genetic engineers modified it to express a specific way when inside the plant. So introduction of host organism is very important. The recombinant DNA is introduced into a host organism such as bacterium or a plant cell using various techniques such as transformation or transfection. So once the gene of interest has been cloned, we know well what to do with this gene. So there are enzymes used to cut the gene of interest. So here one or more of the three gene regions can then be replaced or modified. The gene regions are blocked back, the gene regions are bounded back together and function as a normal gene. Since the DNA has been cut apart and put back together into a new combination, it is called as recombinant DNA, or it is also called as rDNA. So this recombinant DNA is having certain characters or certain traits, and we can incorporate it into the host plant after cloning. After gene modification, the new gene is inserted into a single plant cell using one of the transformation method such as the gene gun or agrobacterium. We know well, in one of my earlier video, I have shown agrobacterium mediated gene transfer. So how gene can be transferred with the help of this technique, that is gene gun. There are different methods of gene gun also. The transgene must land in the cell's nucleus and be incorporated into one of the chromosomes. This enables it to be expressed and passed on to the offsprings. So expression of transgene is the last step. The host organism expresses the transgene, resulting in the production of a new protein or trait. So this step is very important. The plant cells are totipotent, meaning a cell from any part of the plant can divide and multiply into another complete plant. We know well the ability of a single cell or certain cells after cell division, after multiplication of cell, every cell divide and redivide, it multiply and it develop entire organism or entire plant. So this potential power or this ability of a plant cell to develop complete plant, this ability or this potential power is called as totipotency. So every plant cell is having totipotent. By considering this phenomenon, we can get entire plants. So every time the cell replicates and divides, all of the chromosomes are copied, including the new gene, the gene of interest, the rDNA, which we have incorporated into the cell. When this cell multiply, the same time the gene of interest or the rDNA which we have incorporated into the cell, that also get multiplied. So multiplication process, and as a result of which we'll get so many cells. So the result is transgenic plant with the new gene in every one of its cells. So every cell contains new gene and we'll get entire plant. Afterwards, this step is very important, that is backcross breeding. So backcross breeding is then used to move the transgene into a high yielding elite line, and the resulting hybrids are tested and evaluated for release. As far as applications of genetic engineering is concerned, so this is very effective in the field of agriculture. It means that genetic engineering has been used to develop crops that are resistant to pests and diseases and certain environmental stresses. So this technology is also used in medicine. So genetic engineering has been used to develop new medicines such as insulin and human growth hormones. So I have also mentioned that this genetic engineering nowadays, so many people, those who are having biotechnology background, they are using the knowledge of genetic engineering. So genetic engineering has been used to develop the new products such as biofuels and bioplastics. In addition to that, this technology is also effective in gene therapy. So genetic engineering has been used to develop new treatments for genetic disorders such as sickle cell anemia and cystic fibrosis. So all these are the benefits of genetic engineering. So this genetic engineering technology is very effective for future research. We know well Stanley in the year 1973, Paul Berg in the year 1972, Joshua in the year 1946. All these pioneers, they have done a wonderful work regarding the applications of plant genetic engineering. So the final result is a high yielding transgenic hybrids or a variety that expresses a new trait with desired character. Out of millions of cells that undergo the transformation process, only one or two may lead to a marketable hybrid. So in modern genetic engineering, the development of genetic engineering has involved the contributions of many scientists and the field continues to evolve. So modern genetic engineering techniques such as CRISPR technology, gene editing have revolutionized the field and open a new possibilities for basic research, biotechnology and medicine. So I hope I will sum up today's my review of plant genetic engineering, how this technology is helpful for making new hybrids with desired characters. So here initially we require host plant. From host plant we have to select certain cells. From cells we have to reach out to the DNA. We have to target the gene of interest. We have to remove that gene of interest. We have to cut the gene of interest and we can multiply that gene of interest with the help of vectors. And after multiplication, with the help of gene gun, we can incorporate that gene into the desired plant, the targeted plant. And every cell will give us the marketable hybrid in the form of new variety or whatever may be the target which we have decided. So this is all about the plant genetic engineering or overview of genetic engineering.